Dear student, I am Dr. 𝗠𝐨𝐫𝐞, Professor in Biochemistry. So in this video, I will be explaining you Introduction to Enzymes, Classification of Enzymes and Mechanism of Enzyme Action. Enzymes are colloidal thermolabile biological catalysts which are synthesized in our cells and they enhance a rate of reaction. Most enzymes are protein in nature except ribozyme. Ribozyme is an RNA having an enzymatic action. So generally enzymes are named by adding suffix ase to the substrate of the reaction. So example, lactase. As you see here, ase it is added as a suffix there, and most of our enzymes they end as ase, but some of the enzymes they end with a trivial name like trypsin. Trypsin is an enzyme, but you don't see that ase there. So otherwise many enzymes in our body they end as ase. So you can easily identify name of the enzyme if you see ase as the suffix. Now, looking at the names of the enzymes, you can differentiate what is the function of that enzyme and whether it needs ATP, whether it uses a water molecule or whether it uses inorganic phosphate or it incorporates oxygen or uses oxygen as an electron acceptor. All that kind of information we can know if you understand the name of the enzyme. Let's take an example here. The difference between synthase and synthetases. Now the synthases, they do not need ATP, whereas synthetases, they need ATP to synthesize a molecule. Coming to phosphatases and phosphorylases, difference between phosphatase and phosphorylase. Phosphatases, they introduce water molecule and break a molecule, whereas phosphorylase, they introduce inorganic phosphate and break a molecule. Example, we have glycogen phosphorylase. Glycogen phosphorylase introduces inorganic phosphate into the reaction as a nucleophile and break glycogen into glucose 1-phosphate, whereas phosphatase, it simply adds water molecule and break a molecule. It's like a protein phosphatase. Now, oxidases and oxygenases. Oxidases, they use molecular oxygen as an acceptor of electron, whereas oxygenases, they introduce a molecular oxygen into the reaction. So like this, we can use enzyme names to differentiate what are the functions of enzymes. Coming to certain terminologies that we use in enzymes. Substrate. Substrate is the reactant which gets into the reaction and converted into a product. So product, as I just explained to you, it is the outcome of enzymatic reaction. Coming to coenzymes. Coenzymes, these are organic molecules that will bind with an enzyme, and coenzymes are necessary for certain enzymes to attain their optimum function. And example for coenzymes can be, there are multiple, so many examples are there for coenzymes. Majority of the water soluble vitamins act as coenzymes. Out of fat soluble vitamins though, vitamin K is the only fat soluble vitamin that act as coenzyme. And some of the molecules that act as coenzymes are not necessarily vitamins, like lipoic acid act as a coenzyme, but it is not a vitamin. All water soluble vitamins act as coenzymes. Example, vitamin B6, pyridoxine. The active form of pyridoxine is pyridoxal phosphate, and pyridoxal phosphate is a coenzyme for more than 100 of enzymes in our cellular metabolism. Now this particular table will give you the list of coenzymes for enzymes on the left side of the table that you see here. Kinases. Kinases, they use ATP or GTP as the coenzyme. Any enzyme that ends as kinase, like hexokinase, glucokinase, so they use ATP or GTP. Dehydrogenases. Dehydrogenases, generally they prefer to use NAD, that is nicotinamide adenine dinucleotide, or FAD, flavin adenine dinucleotide. Pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, branched chain alpha keto acid dehydrogenases. These are all complex enzymes. They need 5 coenzymes, and they are thymine pyrophosphate, which is TPP, lipoic acid, coenzyme A, which is the derivative of pantothenic acid, FAD, which is a derivative of riboflavin, NAD, which is a derivative of niacin. So these 5 coenzymes are necessary for enzymes that are listed here, that is pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase complex, and branched chain alpha keto acid dehydrogenase complex. Trans-ketolase enzyme. Trans-ketolase enzyme, which is there in pentose phosphate pathway, needs thymine pyrophosphate as the coenzyme. Transaminases, there are 17 transaminases, and all of these transaminases, they need pyridoxal phosphate as the coenzyme. Carboxylases. Carboxylases, they need biotin as the coenzyme. Like this, so we have variety of coenzymes for different enzymatic reactions in our body. Now let's move on to see what are the examples for enzymes that need cofactors. Now the cofactors are inorganic molecules that will bind with an enzyme and enhance the enzyme activity. Now generally the cofactors are metal ions. If the enzyme binds with the metal ion, and if the enzyme activity is increased in the presence of metal ion, we refer that kind of enzymes as metal activated enzymes. If a metal ion strongly binds with the enzyme as a prosthetic group, so we just call it as metalloenzyme. So metal activated enzyme and metalloenzyme. So these are the two names here. You got to differentiate that. Now let's see what all the cofactors, examples for cofactors here. Carbonic anhydrase, alcohol dehydrogenase, aminolevulinate dehydrogenase. All these enzymes, they need zinc as the cofactor. Enolase enzyme, which is there in glycolysis, that needs manganese as the cofactor. Cytochrome oxidase, catalase, peroxidase, these enzymes, they need iron as the cofactor. Xanthine oxidase, which is involved in uric acid synthesis, it needs molybdenum as the cofactor. Salivary amylase enzyme needs chloride as the cofactor, and kinases, they need magnesium as the cofactor. Now let's see what is co-substrate. Co-substrates are generally, these are the coenzymes that can sometimes act as a co-substrate. That means NAD, that is nicotinamide adenine dinucleotide, can get into the reaction and convert it as NADH plus H plus. So NAD simply act as a co-substrate here. NADH plus H plus will be co-product. Similarly, FAD can get into the reaction and come out of the reaction as FADH2. So FAD is a co-substrate, FADH2 is the cofactor. Let me write it down here. So a substrate, when it is converted into a product by an enzyme mediated reaction, so NAD getting into the reaction and come out of the reaction as NADH plus H plus. So here NAD is a co-substrate, NADH plus is a co-product. So this is how a co-substrate and co-product can be differentiated. Now what is a cofactor? Cofactor is a separable form which is binding to an enzyme. So if a metal is attached to an enzyme, it is a metal activated enzyme, as I already explained to you earlier and given some examples. Prosthetic group. Prosthetic group is a non-separable molecule that will bind with an enzyme. That means it is tightly attached to an enzyme and you cannot separate it from the enzyme. And a metal which is totally attached to an enzyme, which is inseparable, that kind of enzymes are called as metalloenzymes. Apoenzyme. Coming to apoenzyme, it is just a protein part of an enzyme without any cofactor, without any coenzyme or without any prosthetic group. Simply only the protein part of an enzyme is called as apoenzyme. Coming to types of enzyme specificities. So enzyme specificities can be classified into absolute specificity, a group specificity, reaction specificity and stereospecificity. So what is absolute specificity here? Absolute specificity is when an enzyme acts only on one substrate and catalyze only one reaction. That means this is absolutely specific for that particular substrate and that particular reaction. Example, we have glucokinase. Glucokinase is absolutely specific for the conversion of glucose into glucose 6-phosphate. But it cannot act on fructose, it cannot act on mannose. So it just only acts on glucose. That kind of enzymes, that kind of reactions, we refer that as an absolute specificity enzyme or as enzyme having absolute specificity there. Coming to group specificity. Group specificity is when an enzyme acts on a specific bond or a group of substrate. Example, we have say glucose, galactose, fructose, mannose. All these are 6-carbon containing molecules. We call them as hexoses. Now, there is an enzyme called hexokinase. So, this hexokinase catalyzes all hexoses by adding phosphate. Like glucose converted to glucose 6-phosphate by hexokinase, fructose is converted to fructose 6-phosphate by hexokinase, mannose is converted to mannose 6-phosphate by hexokinase. So, that means all hexoses are catalyzed by the same enzyme called hexokinase. So, hexokinase has the group specificity there. Similarly, phosphatases. Phosphatases, they hydrolyze organic phosphate. Anything that has attached with the phosphate, it can be removed by adding water. And example is protein phosphatase enzyme. Irrespective of which protein has attached to the phosphate, simply protein phosphatases add water and remove that phosphate from the molecule. Coming to reaction specificity. Reaction specificity is enzymes are specific for a particular reaction even though the substrate is same for each reaction. Example, a conversion of pyruvate into different products. So let me write down that so that we understand exactly how a reaction specificity goes. So pyruvate is a central molecule now for us. Pyruvate, and this pyruvate can go into acetyl-CoA formation. This is done by pyruvate dehydrogenase complex. Pyruvate can go into oxaloacetate formation. Pyruvate can go into lactate formation. Like this, so there are different ways that pyruvate can go into. So, for each of these different direction, we have a specific enzymes. Like conversion of pyruvate into acetyl-CoA, it will be done by enzyme pyruvate dehydrogenase complex. Conversion of pyruvate into oxaloacetate, it is done by an enzyme pyruvate carboxylase enzyme. Conversion of pyruvate into lactate, it is done by lactate dehydrogenase enzyme. So like this, pyruvate can go into different direction. Even pyruvate can also be converted into alanine. So this conversion of pyruvate into alanine, it will be done by enzyme alanine transaminase. So now you have seen that pyruvate is the central molecule, and this pyruvate is going into different direction by a different enzyme, although the substrate is same. So this type of specificity we refer it as reaction specificity. Enzymes are very specific to the reaction that they catalyze, although the substrate may be same. Coming to our last specificity here, that is the stereospecificity. Stereospecificity means enzymes act only on one type of stereoisomer. Example, we have L-amino acid oxidase, which will act only on L-amino acids, where we have D-amino acid oxidase, which will act only on D-amino acids. So these are all the different types of enzyme specificity, that is absolute specificity, group specificity, reaction specificity, and stereospecificity. Now let's look at classification of enzymes. Since we have hundreds and hundreds of enzymes in our metabolic pathways, so there is a systematic way to classify these enzymes, and this classification, it is done by Enzyme Commission of IUBMB, which has given a systematic name, which is a unique 4-digit identification number given for every enzyme. Now how this 4-digit identification number is given here? So that it is referred as EC number, EC for Enzyme Commission number. So it is based on the classification of enzymes. So the very first class of enzyme example is oxidoreductase class of enzyme, and that oxidoreductase, since it is number 1 in the classification, it is the number 1 class there. So it is given as number 1 in the beginning. So 1 indicates the class of enzyme there. And then second number besides the first number is the subclass of that particular enzyme. And that subclass usually indicates the type of substrate that is getting into the reaction. And the third number is the subclass of an enzyme, and that indicates the type of cofactor that this particular enzyme needs. And the fourth number is a specific enzyme within the subclass. So like this, an enzyme action is divided into different ways, different categories, and thereby we can easily identify by knowing that unique 4-digit number given by Enzyme Commission. Okay, so the number 1 in that 4-digit is the class of an enzyme, number 2 indicates subclass of an enzyme, number 3-digit indicates subclass of an enzyme, number 4-digit indicates a specific enzyme within that subclass. Now let's look at classification of enzymes. Enzymes are classified as oxidoreductases, transferases, hydrolases, liases, isomerases, and ligases. Our very first class of an enzyme here is oxidoreductase class of enzyme. As the name says, oxidoreductase class of an enzyme, so they will catalyze oxidation reduction reaction. As you know, oxidation is the loss of electron and reduction is the gain of electron. So whenever a loss of electron or gain of electrons are going on in a metabolic reactions, or when a substrate is converted into product, so that type of reactions or the enzymes that are catalyzing this type of reaction are simply grouped under oxidoreductase class of enzymes. Examples, how the oxidoreductase class of enzymes will end. So oxidoreductase class of enzymes, generally they will end as oxidases, dehydrogenases, peroxidases, oxygenases, reductases. Look for these names at the end of enzymes. Oxidases, so they will use oxygen as an electron acceptor. Dehydrogenases involved in oxidation reduction reaction, where there is a loss of electron and gain of electron, usually coupled reaction. Peroxidases, where hydrogen peroxide is neutralized into a water molecule, something like that. Oxygenases, incorporation of molecular oxygen into the reaction. Reductases. So, any enzyme that ends as these oxidase, reductase, dehydrogenase, oxygenase, peroxidase, you simply put that into oxidoreductase class of enzyme. Now, coming to our second class. Second class of enzyme is transferases. Transferases, simply they transfer a group other than hydrogen. And transferases, they generally end as kinases like hexokinase, glucokinase, protein kinase. And transferases, some of the transferases are transketolase, transfer of keto group, transaldolase, transfer of aldehyde group, phosphorylase, inorganic phosphate is incorporated and basically it is transferred to a molecule and say glycogen breaks into glucose 1-phosphate. So, inorganic phosphate is transferred there. Glycogen synthase where glycogen is synthesized, like glucose is transferred. So, like this transferases, say basically transfer a group other than the hydrogen. Hydrolases, coming to hydrolase, our third class of enzyme is hydrolases. Hydrolases, they catalyze hydrolysis of certain bonds. What kind of bonds hydrolases can break? Ester bond, ether bond, peptide bond, glycosidic bond. This kind of bonds are broken by adding water. Hydrolysis, addition of water and breaking a bond. Example, all digestive enzymes present in the intestinal lumen, they are all hydrolase category of enzyme. Phosphatases add in water and separate a phosphate. So, these are the hydrolases. Coming to our fourth category, it is lyases. Lyases, they catalyze a removal of groups or break bond without adding water. So, that's the difference between hydrolases and lyases. Hydrolases, they will add water and break a molecule, whereas lyases, they do not add water but still break a molecule. Example for lyase is aldolase, which comes in glycolysis, fructose metabolism. Decarboxylase, removal of carbon dioxide from an organic molecule, like histidine decarboxylase, where histidine is converted to histamine. Tyrosine decarboxylase, tyrosine into tyramine. Glutamate decarboxylase, glutamate to GABA. So, these are the decarboxylase which belongs to lyase class of enzyme. Then synthases. Synthases are lyase class of enzyme. So, like glycogen synthesis of glycogen, right? So, and then hydratases, they will add water, right? And desulfidrases. So, these are the examples of enzymes that belong to lyase class. Now, looking at the fifth class of enzyme is isomerase. Isomerases, they catalyze optical, positional, geometrical isomerization of substrate. Example, they end as isomerase. Any enzyme that ends as isomerase, of course, belong to class of isomerase. Epimerase, enzymes that are participating in epimerization reaction end as epimerase. Mutases, racemases, enzymes participating in racemization reaction. So, isomerases, epimerases, mutases, racemases, they are all isomerase class of enzymes. Then ligases, our last class of enzyme is ligases. They catalyze binding of two substrates by using energy, and usually that energy is ATP. Examples for ligases, we have carboxylase. Carboxylases are ligases. Example, pyruvate carboxylase, conversion of pyruvate into oxaloacetate, where carbon dioxide is added into a pyruvate to make oxaloacetate. Or acetyl-CoA carboxylase, conversion of acetyl-CoA, 2-carbon acetyl-CoA into 3-carbon malonyl-CoA by adding carbon dioxide. So, let me write that down. Acetyl-CoA is a 2-carbon molecule which will be converted into melanyl-CoA. Melanyl-CoA is a 3-carbon molecule. That means that additional carbon, it will come from carbon dioxide, and for that ATP is needed. So these are ligases. So carboxylases, they need ATP to add carbon dioxide, and also they need magnesium. So that type of enzymes are called as ligases. Synthetases are also ligases, because synthetases, they will add an organic molecule to increase the size of the molecule, like glutamine synthetase. Glutamine synthetase, where ammonium ion is added to glutamate to make glutamine by using ATP. That's the example for synthetase, like ligases there. Now let's move on to see how does enzyme work. What is the mechanism of enzyme catalysis? How exactly enzyme works? Because enzyme is a small molecular weight protein. Most of the enzymes are protein in nature. So they will bind with the reactant, that is the substrate, and convert that substrate into product. But at the end of the reaction, enzyme comes out unaltered. Basically what it does is, it is simply converting substrate into product without being altered by itself. So the enzyme action can be explained by two models. One is a Fisher's lock and key model. In Fisher's lock and key model, it is the enzyme, it has got a complementary shape or a structure to a product and the substrate there. So where substrate will go and bind, and it is converted to product, and product is released. It's a fixed model. Fisher's lock and key model is a fixed model, so it doesn't change. It's like a lock and key. Okay, so this model does not explain all the enzymes activity in our body, because in our cells, majority of enzymes, they don't have a fixed shape which is complementary to the substrate that is binding to it. So lock and key model is a kind of outdated model, because it do not explain exactly how the mechanism is going on in our body. Now the model that is accepted and which explains the mechanism of enzymes, or most of the enzymes, how they work in our body, is the 𝐒𝐲𝐿and's induced fit model. The 𝐒𝐲𝐿and's induced fit model, what it shows here is, enzyme, the shape of an enzyme where the substrate is going to bind, may not be exact complementarity to the substrate shape. As you see here, the A, B and C in the active site of an enzyme, and look at the complementary shape of a substrate which is coming and binding to the active site. So it is not exact complementarity here. So how this enzyme substrate will bind to an enzyme? So whenever substrate will come and make an initial contact with an enzyme's active site, like suppose it is making an initial contact with A. So once that initial contact happens, like once it initiates a bond formation there, so enzyme itself will undergo conformational change, so that it will well accommodate the substrate and fit that substrate well. So basically there will be conformational change that is going on in the overall protein structure of, particularly at the active site of an enzyme, so that substrate is well accommodated and more bonds are formed between the substrate and the enzyme. And that type of dynamic model is explained by 𝐒𝐲𝐿and, and it is referred as 𝐒𝐲𝐿and's induced fit model. And this is the model that can explain how our body enzymes work, and this is the most accepted model for enzyme action, mechanism of enzyme action. Now, where exactly the action is going on when when the substrate is converted into product? So every enzyme has got an active site, and this active site, it is a special pocket or a cleft or a crevice in an enzyme where a substrate will go and bind and be converted into product. So whenever the substrate binds to an active site of an enzyme, makes an initial contact with the active site of an enzyme, so as I told you earlier, that it is going to make a conformational change in the active site of an enzyme, thereby substrate is making more and more bonds with the active site of an enzyme and it is going to fit well. And when it fits well, so that initiates the product formation. So this particular mechanism is explained by Coastland by his Coastland's induced fit model, where there is it induces a conformational change in the enzyme's active site, and that allows the catalysis. Here, as you see, a substrate which is coming and binding to an active site in the number 1, you see in the number 1. So it shows a substrate making a contact with the active site of an enzyme. And when it makes an initial contact with an active site of an enzyme, so only few bonds are made there, and it induces a conformational change where enzyme-substrate complex induces a conformational change in the active site of an enzyme. Once it induces a conformational change there, additional bonds are formed, and that makes a proper fitting of substrate with an active site of an enzyme, because we need all the additional bonds there, so that more and more bond formation will take that substrate into its highest energy state called transition state. And this is the requirement for substrate to go into product. And product is formed and enzyme is coming out of the reaction unaltered. So what happens here is, it is the active site of an enzyme where substrate is going and binding. And in that active site, near to that active site, there can be cofactors, coenzymes and all that. So initially, substrate is making initial bonds or the connection with the active site of an enzyme. And because of that, there will be conformational change and more bonds are made. And that will take a substrate into its highest energy state called transition state. And in this transition state complex, because it is most unstable, it will go into product formation, and the products are released. Enzyme will come out of the reaction unaltered, and that is now the enzyme is ready for next reaction based on the specificity of the enzyme. Now look at the graph here. So there are two lines here. One is whenever a substrate is converted into product without any enzyme mediated action. That's the red line you are seeing here. So substrate is initially converted into substrate transition state, and from substrate transition state it falls into a product. So the amount of energy that is needed for the substrate to go into transition state is shown with the red bar here. And look at the length of the red bar. So it is higher. So that is the energy which is needed for a substrate to go into its transition state. So when substrate is reaching ST for transition state, so it is the highest energy state of the substrate and it is the most unstable state, and it falls into a product which is the lower energy state there. So basically substrate from higher energy state, it is going into a product which is at lower energy state, and before it is becoming product, it is reaching at highest energy, that is the substrate transition, and the amount of energy that is needed to take substrate into its transition state, we refer that as free energy of activation. Free energy. Free energy of activation, and this free energy of activation is the one that will take substrate into transition state and that facilitate the product formation. This happens in the absence of enzyme. Only thing is the free energy of activation that is needed there is much higher. That's why the conversion of substrate into product, it slows down. It takes a lot of time. Now let's see what our enzymes will do. Enzymes will bind with the substrate to make enzyme-substrate complex, as you see here in the green line in the figure. So enzyme-substrate enzyme-substrate complex, so where the initial bond formation is going on, and that will lead to a conformational change in the active site of an enzyme, making more and more additional bonds as we have seen in the previous slide, and that will take the substrate into transition state much earlier, because the binding additional bonds that are forming between enzyme and substrate will actually release energy called as binding energy, and this binding energy will actually bring down the amount of energy that is needed to take substrate into its active state, into its transition state, and that you see in the green bar on the right hand side, that the free energy of activation in the presence of enzyme is much lower than the free energy of activation in the absence of enzyme, that you now can see in the red bar there on the left hand side. So the amount of free energy that is needed for a substrate to go into transition state in the absence of enzyme is much higher compared to an enzyme substrate complex going into enzyme substrate transition state. The free energy of activation is much lower, and why it is much lower? Because the binding energy that is released because of the additional bond formation going on between enzyme and substrate complex is decreasing that transition free energy of activation, thereby it enhances the rate of reaction. So the reaction goes on much quicker than in the absence of enzyme. So enzyme substrate transition state goes into enzyme product, and the product is separated, enzyme comes out of the reaction unaltered. Now let's look at the similar graphical representation here. So the blue line that you are seeing in this particular figure is an uncatalyzed reaction in the absence of enzyme, and the dotted green lines that you see here, dashed green line that you are seeing here, it is the enzyme catalyzed reaction, that is the catalyzed, it is written in the bracket. Now look at the conversion of substrate into transition state in uncatalyzed reaction, and look at the amount of free energy of activation that is necessary to take substrate into transition state. The big arrow that you are seeing here, that is much bigger, indicating that free energy of activation is much higher. Whereas look at the dashed green line, it is a small arrow indicating that free energy of activation is much lower. That means enzymes simply decreases free energy of activation, which is denoted as delta G plus plus. You got to read it like that, delta G, and write 1 plus and put another plus on top of it. It's a delta G plus plus. It's a symbol for free energy of activation, or simply called as activation energy. So what enzymes simply do is they lower the activation energy, thereby enhance the rate of reaction. Now see the energy difference between the substrate and the product. So the energy difference between the substrate and the product, when you compare with the catalyzed and uncatalyzed reaction, it doesn't change, because both catalyzed and uncatalyzed reaction, so the energy of the substrate and the energy of the product, they are at the same level. So that means there is no change in the energy of substrate and energy of product between catalyzed and uncatalyzed reaction. That's why delta G is the Gibbs free energy. Gibbs free energy, it remains same whether it is a catalyzed reaction or uncatalyzed reaction. So just to put it in other words, like enzyme catalyzed reaction decreases delta G plus plus, whereas delta G remains same. And this is what happens in an enzyme catalyzed reaction. This is the mechanism of enzyme catalysis. So enzymes simply, they lower the free energy of activation, that is delta G plus plus, but they do not alter overall energy of the reaction, that is delta G, or the Gibbs free energy do not change. So look at that, what I have explained in the previous slide. It is written here very clear, enzymes do not alter the overall energy of the reaction. So that means Gibbs free energy, it remains same, delta G remains same, or the energy difference between the substrate or products remains same. So what enzymes only do is they only decrease the energy of activation or the delta G plus plus. Now the table here, it is depicting the energy and the rate of reaction. Delta G is not affected by enzyme catalyzed reaction, whereas enzymes, they simply increase the rate of the reaction, conversion of substrate into product. How enzymes will increase the rate of reaction? Because enzymes will simply decrease the free energy of activation, that is delta G plus plus, thereby they enhance the rate of reaction. So now based on the value of delta G, if the delta G of the reaction is less than 0, it's a thermodynamically spontaneous reaction, energy is released into the system, and most of the time this kind of reactions are irreversible and usually they are rate limiting reactions. If delta G value is more than 0, that is in positive sign, it's thermodynamically non-spontaneous reaction. Energy is required for the reaction to go on. They are endergonic reactions. If delta G is equals to 0, reaction is at equilibrium. Usually these are free reversible reaction, freely reversible. Okay, so these are some of the meanings for delta G based on the delta G values. So that's all about general introduction to enzymes, some of the terminologies that are used in enzyme catalyzed reaction, mechanism of enzyme action.